So started with the carbohydrates because they are, as I have already told you, they are the most important metabolic metabolites. They are the most important compounds or macromolecules involved in the metabolism. So first of all, of course, for the carbohydrate metabolism, you have to have glucose, and glucose have to flow or move from the blood into the liver. And for that, there are certain transporters known as GLU2, glucose transporter 2. These transporters, they are, specifically made up of liver. They are not sensitive to insulin. So insulin don't affect the functioning of these transporters. So in this way, we can say that regulation of the transportation of glucose into the liver is not as much regulated as it is regulated in other organs. And it is mostly the concentration based. If there is more concentration of the glucose in the blood, it will definitely go into the liver, which will be utilized in the formation of glycogen. And if it's not much needed, if there is no much glucose into the blood, then it will be degraded. The glycogen will be degraded and given rise to the glucose, which will be then used by the body. Glucose phosphorylation is a very important thing. It's an interesting thing, you have to understand it. The phosphorylation of glucose, you all know, through the activity of the enzyme hexokinase, it gives rise to the formation of glucose 6 phosphate. And the formation of glucose 6 phosphate is very much regulated by insulin, because we know that insulin is very much involved in the regulation of the amount of glucose into your blood. So if once glucose gets phosphorylated, giving rise to the formation of glucose 6 phosphate, it gets trapped into your liver. What does this mean? It gets trapped, that it cannot go out then. Why? Because there is no effective transporter for the transportation of glucose 6 phosphate in the liver. So as a result of this trapping of the glucose 6 phosphate into the liver, what happens that now glucose is available for further metabolic pathways, either for the formation of, for instance, glycogen, or then it can also be converted back into the glucose, to the glucose. If you remember the metabolism of glucose, you remember the glycolysis, the final product of the glycolysis was the pyruvate. Fine. Now, the pyruvate then can give rise to the formation of the lactate, if we are talking about anaerobic glycolysis. But in the aerobic glycolysis, the pyruvate will be converted into the acetyl CoA, which can then either go towards the formation of lipids or can become the part of the Krebs cycle. Now, also you should keep in mind that excess glucose storage also help in the formation of fats. Why? Because if there is so much excess of the glucose which is giving rise to the formation of glycogen, ultimately then when the body doesn't need, or when the liver doesn't have much capacity remaining in it for the formation of, for the storing of more of the glucose, what will be happening, that then it will, rather being converted into the glycogen, it will be used in the formation of lipids. The lipids are formed because of the conversion of pyruvate into the acetyl CoA, and then acetyl CoA is the major unit of the fatty acids. Also, you should keep in mind that one of the major function of liver is the control the concentration of glucose, to control the level of glucose in your blood. So if, for instance, under certain circumstances when your body is deprived of glucose, there is much less glucose, what will happen, that then either the glycogen will be getting degraded, giving rise to the glucose, or you will start getting glucose from the non-carbohydrate moieties by reaction, you all know, by the process you all know, the gluconeogenesis. Again, whether it's the formation of glucose from the degradation of glycogen or whether it is the formation of glucose by the process of gluconeogenesis, in either way, liver have very important and fundamental role to play. Once the glucose, your body needs glucose, then glucose has to be moved out of the liver. What will happen then? Glucose 6 phosphate is acted upon by the phosphatase. The phosphatase will then free glucose from the glucose 6 phosphate because of the dephosphorylation of glucose 6 phosphate, and this glucose then will be used by the body.